My name is Daniela Munezero. I'm a PhD student at West Virginia University, and today I'm going to share with you how our eyes function to form an image for us to see. Imagine how hard it would be not being able to see the beauty of the sunset, or the beauty of the nature around you, or simply how great you look in your favorite outfit. Well, that's the experience of our friends and family who are or have already lost their sight. And as we all know, sight is the dominant sense for people who are able to see, as we rely on our vision in our daily work. And it's in that case that my research is dedicated on understanding how our eyes function in the formation of the image that you and me are seeing right now, and more importantly, how we can use that knowledge to finding new ways or new approaches to treat blindness diseases. Today, I'm going to share with you the basic process by which our eyes function in the image processing. I will then also share specifically on one part of the eye that my research focuses on, called rhodopsin, which is involved in the sensing and capturing of the light. I will wrap up my presentation by showing you how the understanding of how the vision process is regulated is crucial to finding new approaches to prevent and treat blindness diseases. Have you ever wondered why you can't see anything at all in the darkness? That's because our eyes relies on the light that bounce back from the object and use it to interpret it into the finite image. When the light hits our eyes, it is sent back to a layer in the back of the eyes called retina. This layer is made of small pieces or light receivers cells that we call photoreceptors. The photoreceptors or the light receivers comes into two categories. We have rods and cones. Our eyes would use rods at night when there is low light intensity, and during the day when there is high light intensity, our eyes are using cones. And more importantly, cones helps us to see different colors, and that's why when it's night it's hard to differentiate or to figure out the color of a given object. And in addition to that, cones and rods are very crucial because they help in the transformation of the light into a form that can be sent and interpreted by the brain into the finite image. My research focuses again more deeply on a small part of the cones and rods that is involved in the sensing and capturing of the light, and we call that small part rhodopsin. Think of rhodopsin as a window through which light can enter through the house, in this case the house being the cone and rods. Once the light is captured by rhodopsin, it is transformed and sent to the brain. After the light has been sent to the brain, this window is temporarily closed. And once the brain has already finished to interpret the image, these cones need to be, or this rhodopsin need to be reopened again. It's in that case that my research aims on identifying how this window get open again by finding new ways or the process by which this window gets open again. And why do I care about that? So as you see, if this window, particular window to rhodopsin is not able to be reopened again after the image interpretation, our eyes will not be able to let in more light. And as we don't have more light, the cones and rods stop functioning. And as these cells, cones and rods are not functioning, they start dying off. And as they die off, this eventually lead to the complete loss of sight. An example is a disease called retinitis pigmentosa. Retinitis pigmentosa is a disease characterized by the progressive death of the light receivers or progressive death of rods and cones. And in patient, as the time goes on, as the cells dies off, the patient progressively lose their sight until they have complete blindness. Our aim is to understand how our eyes function to regulate the vision processes, and to use this knowledge or this understanding to finding new ways or new approaches to design therapies and treatment for blindness diseases. Understanding of how our eyes regulate the image processing helps us to have a link between the blindness diseases and the defect in the vision processes, and we use that to design new therapies that can correct any of those defects, and this will allow us to treat and prevent blindness diseases. As I end my presentation, I hope that everyone in the room has a better understanding of how our eyes function to mediate or to help us process the image and allow us to see. And as I continue in my research, I hope to work with other researchers, and also by the financial help of our community and government, to find new ways to treat blindness diseases so that no one has to lose sight again. With that, I thank you for your undivided attention and for giving me this opportunity to share my research today. And with that, I'm happy to take any question. Thank you.